Dr. Wrobel, my name is Dr. David Seftel.
Hello, David.
I'm a medical doctor and the Director of Research and Development for the Macular Degeneration Foundation. Today we're very privileged to have you here to talk about the development of a potentially new and breakthrough therapy for RP patients and perhaps other patients with retinal degeneration. You have a very, very extraordinary career path with some of the finest names in ophthalmology from a technological perspective. So please tell us how you came to be involved with this particular adventure in technology and ophthalmology.
Well, first I have to explain how this project started in Germany back in the 1990s. The first ideas came up that it should be possible to replace lost photoreceptors with camera chips. And there was a famous German newspaper writing an article and quoting a famous German electrophysiologist, quoting him saying that the idea to restore vision with a camera chip is as remote as flying with the space shuttle to the next star. And this guy doesn't want to be remembered about this statement because a few years later he started a research project funded by the federal government. And later on, when the federal government decided that basic research has been done, he was asked to find a strategic investor, namely an important company funding the development of a product. And in this respect, he contacted me. I was at this time Chief Technology Officer of Carl Zeiss Meditech, one of the big names here on the floor. And I sent someone to look into the project, wrote a report, suggested to my fellow board members that we should, Carl Zeiss Meditech should enter this project, and the result was very disappointing. Oh, 7 million? That will destroy our next quarterly result. And break even 3 years? Oh no, that's much too long. And 3 years was, well, we know it was very optimistic. Anyway, I was disappointed, and because I was convinced that this project is going into the right direction, it's one of the few projects where I thought it has a big future. So I was quite unhappy. And then in 2004, I went to Professor Retina and telling him that, well, if I would like to help you, but my impression is it's wonderful research. What you need now is a professional from industry. And I know that you cannot afford to pay me a salary right now, but perhaps we can talk about shares. And so I joined the company in September 2004 as CEO. And my first job was to look for investors. And what I didn't say up to now is Professor Retina didn't find a strategic investor. It was too risky for every established company. So the only choice he had was to found his own company with some experts who have been involved in this research project. And that was basically a company with only a box office and nothing else when I joined the company. So that's the way it started.
So you grew it from a seed, essentially.
Yeah. We started with 4 people, and now we are, I think, 22.
Excellent. So tell us a little bit about the device that you have CE approval for in Europe. What is it used
We do not have yet CE approval. We're working on that. Okay. The device is a subretinal chip. That's the big difference to other approaches. Retina is a retinal physiologist, so he understands very well the structure of the retina, and he was of the opinion that the best place or the only place to position a camera chip in the eye is below the subretinal space, because in this subretinally the photoreceptors are there, they are degenerating, they are not there anymore, so they have to be replaced. So you put a camera chip into this area, and then you have a one-to-one correspondence between photodiodes absorbing the light, amplification, and electrodes more or less at the same position. So where there is light, there will be electrical excitation. It's a one-to-one correspondence, and the phosphanes, the electrically excited percepts which the patient is seeing are moving with the eye. So it's eye-hand coordination is still possible. And that was the basic idea, and it originally started with animal experiments, in vitro experiments on retinal samples, what currents you need to stimulate the retina, what voltages and so on, how do you design the electrodes and so on. And that was done until 2005, and 2005 was then the first clinical trial being done in Germany, where we have put in a chip in the human eye for the first time. And at this time, the clinical trial protocol said 1 month, then it should be explanted. And the patient was seeing some visual percepts. He was even able to see horizontal structures, vertical structures, but not much more than that. So in one case, we were extremely happy that the idea of a subretinal device really worked. On the other hand, we were a little bit disappointed that the results were not better. And then we worked on that, did many investigations, were investigating the chip. Does it really work as we expected? And Retina was looking into the problem, does the retina work as we expected, and so on. And finally, in 2008, we had a patient from Finland where we thought that the only problem which we couldn't solve up to now was, is it really true that you do not need to have the chip directly under the fovea, but outside of the fovea? Because, you know, the chip doesn't have such a high resolution that you can restore normal vision. So basically, we think it doesn't make much sense to put it directly under the fovea. And this assumption was proven to be untrue with this patient from Finland. He was the first one, the last one in the pilot study, but the first one who got the implant directly under the fovea, and we were totally surprised by his visual percepts. He was able to read letters. He was able to identify a banana on the table, point onto it. He was reading his name and telling us that there was a error in the spelling of his name. And he was counting his fingers, looking at his hand. He was looking around in the room and could tell that there are so many persons standing around, large, small, and so on. So that was for us a real breakthrough.
So what is the current status of the retinal implant project?
We have now, after the pilot study, which ended with this Finnish patient, we have been using a new device which can be implanted permanently. So patients are allowed to take the device at home, use it at home on their own. And in the first phase, since May 2010, 10 patients have been operated in Tübingen, Retina, close to our company headquarters. And since this year, we have expanded the trial in the multi-center phase, with international implantations being done in Oxford, London, Hong Kong, with Budapest, Hungary, Dresden, Germany, and Singapore joining the next couple of weeks.
How is this device powered?
It needs an external power supply, because it turned out that the human retina is much more sensitive than any camera chip. So we need an outside power supply. And the pilot study was done by a cable running transcutaneously to a point behind the ear where it was passing the skin, but that was it's not a permanent solution, because there is always the danger of infection. And the second study, which was started in 2010, we have a wireless power supply, which means that there is a ceramic package behind the ear covered by skin. The ceramic package contains some electronics, an induction coil, and a permanent magnet. And you put another permanent magnet and a coil on the skin. It fits to the skin, and then there is a cable going to a cigarette-size power supply with some batteries in it, and 2 knobs where the patient can himself adjust brightness and contrast. And contrast is important for adapting to different lighting conditions. So if the patient would leave, for example, this room with artificial light, go outside into bright sunlight, he would stop at the door and readjust so that there is a lot of contrast in the image he is perceiving.
How much power does it consume? In other words, how often does it need to be charged? Does it have an internal battery, or does it require?
No, it has a standard battery, and the batteries have to be exchanged after a few days. But consumer battery is quite cheap, and there is no internal power supply implanted.
So now, from this particular project, you then became involved in transcorneal stimulation.
Yeah.
So tell us about that evolution and what the hopes are in that area.
Yeah, that was a very unexpected spin-off of our basic research into the long-term results of electrical stimulation. We had asked an institute in our neighborhood to do this basic research, and suddenly I received a big report about it, and other people told me, you don't have to read that, it's so thick and it's not so important. And I've read it and I didn't understand anything because there were many foreign words in it, which I didn't know at this time, glia cells, apoptosis and so on. And so I went to the author and asked him, explain it to me. So that was the last chance before putting it into the wastebasket. And he was trying to explain it to me, and after half an hour, I think we were coming to a situation where he said, well, the electrical stimulation induces cell changes. And the strange thing is that these cell changes are normally considered to be indicators of healing effects. I said, could that mean that with electrical stimulation you could retard retinal degeneration? And he said, yeah, that could mean it. And then I said, we have to do something. And so I looked into the existing literature, which has been there for quite a long time, and turned out that never a sound clinical study was done with electrical stimulation. So there were anecdotally reports about miracles of healing, and all the other patients where it didn't work were not mentioned. So I thought, if you have a certain effect, then probably in the last decades that the effect was masked by unprofessional clinical trials. So the critical point is having a clinical trial. And we have found in our major investors someone who is really interested in that and who thinks that such a preventive treatment of retinal degeneration is even more important than a retinal implant. So he was ready to fund that. And the basic idea was that we have to adjust the level of stimulation in such a way that we are really generating phosphanes. And we have known from the Retina implant study that the currents you need to generate phosphanes differ very much from patient to patient. So, first of all, we have to measure the individual threshold current of the patient. Then we adjusted it for stimulation to 150%, two control groups, 170%, one no current. And we did a clinical study with 24 patients, and it turned out the 150% group has shown a statistically significant increase in visual field area by 20%, a trend towards better visual acuity, and the other control groups didn't show any improvement. So there was an effect. And based on that, we have developed a device which can be used in home care, and got CE approval for that. And right now it's being tested further in a post-marketing study in Europe, which we call TESOLAR, with more than 100 patients and 5 different sites in Germany, one in Norway, one in Denmark, Oxford and London joining shortly. And we're also working with Wills Eye Hospital, Philadelphia, to start a study here in the US.
Now, your initial thresholds, that was done with ERG, I assume. Or how did you determine, what techniques do you do to determine?
Well, the traditional method to determine the individual threshold was to fix a silver-coated nylon thread to the surface of the cornea. You lay it on the surface of the cornea, you fix it with adhesive tape, and then you have a power supply where you can adjust the current levels and increase them, and then the patient at a certain current level says, oh, I can see something. It's flashing white. And that's the threshold current. And all the stimulation was done with the same scheme. Silver-coated threads fixed to the face. And for a home care device, of course, it has to be different. It has to be much easier. So the arrangement now is that we are using some kind of disposable loop, still a nylon thread, which you put into a spectacle frame, and the spectacle frame has to be once adjusted to the patient. The patient himself, who can be blind in principle, puts in this loop, this nylon loop, then opens his eyes, puts his spectacle frames on, and closes the eyes. And then he presses a button on the power supply. Then the power supply is stimulating him for the time and with a strength adjusted by the ophthalmologist before.
How many times a day does the patient have to do that?
The original stimulation was done in the first study, 30 minutes per week, once per week, and this is what's now being done in this open label study. We also have another second clinical study going on in Tübingen, where with a control group, double-blinded, where this parameter range is extended. Because you might say if 150% is effective, what about 200%? Yeah.
So are you trying to develop a dose-response curve, or do you now have an optimal dose and frequency?
Well, the problem is that you can have very different parameters. You can vary the time duration, you can vary the repetition rate, and so on. And in order to prove any benefit, you need patience. And so it's quite complicated to investigate this whole parameter range. So we are trying to extend the parameter range step by step. And in the ongoing clinical study, we have 58 patients for this 200% testing. And in the open label study, we will have more than 100% patients all over Europe. So the parameter range may be extended, but quite slowly, because it needs such a lot of effort, and having 50 patients for a clinical study in retinitis pigmentosa is not easy.
Not easy.
Yeah.
Now, you're obviously stimulating the cornea. Is there any difference between stimulating the cornea and stimulating the periocular area in terms of getting the electrical stimulation to the retina?
The reason why we are stimulating the cornea is that, because the cornea is wet as well as the lid rims. So the electrical contact is much more reproducible compared to, let's say, the skin, where the conductance depends on fat in the skin, or when did you last clean it, with soap or without soap. So that's quite unreproducible. And we depend on a specific electrical threshold. So with contacting the cornea, it's much more reproducible.
But isn't there a compliance issue potentially, particularly with older patients who may not be able to?
There is a compliance issue. The threshold current very much depends on the state of the generation. If the generation has been going on for decades, then the current may be so high that it generates pain, and then we cannot use this therapy. So it's directed at patients who are still able to see, but who have the first visual defects, and they want to do something against that.
Now, what type of current are you actually using?
We are using a biphasic current with the hertz repetition rate, 20 pulses per second, and the positive and the negative phase are always 5 milliseconds.
And how did you decide that?
Well, we were looking into the literature. Animal experiments have been done, especially by some Japanese groups. And we looked at the parameters which have been used most and had some effect, at least in animal experiments. So we came to this parameter range.
Now you say you're starting at Wills, a clinical trial. Are you in the recruitment phase for patients at this time?
Well, the Wills has sent a pre-IDE submission to the FDA, is now in talks with the FDA how to do it, and they first, of course, need approval from the FDA for this clinical study.
Because we, at the Macular Foundation, one of the services that we provide is we update patients with upcoming clinical trials that they may want to be candidates for. So I assume your trial will be listed on the clinicaltrials.gov website, and that information will be available. So if people are interested in potentially being candidates, they should go to those resources, or is there an additional contact that you have here?
No, we are closely working with Wills Eye, and there is no other contact I can offer.
And this device, as it is going to be, this device is CE approved in Germany since 2011.
December 2010. Yeah.
Are there phase 4 clinical trials going on in the market with regard to the ongoing efficacy of this device?
Well, what I mentioned, this TESOLA study, this open label study, is a post-marketing study in this sense. It doesn't have a control group because this would be unethical, but the patients are coming regularly to the ophthalmologist, and there is a review. Are there any positive results, or are there any risks? So this is a typical post-marketing study.
And that data is going to be published?
Of course, yeah. We hope that we will have another publication with many more patients in the second half of next year.
And in terms of reimbursement, obviously in Germany it is paid for by the...
Not yet, because if you have a treatment which can be used in ambulatory care in Germany, you need years to get a reimbursement code. So it has to be until the time when we get this reimbursement code paid for by the patients themselves.
And are there any disposables involved in this device?
Well, the nylon thread is with its holder it's a disposable, and it comes ethylene oxide sterilized.
So the treatment is essentially given once a week for 30 minutes. And have you measured the persistence over time? The persistence of the effect. If you stopped giving a treatment, how long does it take for the effect to decay?
Well, in the pilot study, which I mentioned, there was 6 weeks after the last stimulation, another visit with the ophthalmologist, and it turned out that this positive effect has been there even after 6 weeks. That was the limitation at this time, and now in the ongoing clinical study, this post-op control period has been extended.
Now, you made the decision to do one treatment per week. How did you come to that decision rather than once a day or once every 8 hours?
That was not our decision. It was a decision by the principal investigators who said, well, let's start, but first of all, once per week and not faster.
Because it's interesting, if you look at the literature in this area, most people are doing it more frequently. And obviously, increased frequency carries risk, but it also potentially carries benefit. So are there plans to look at again, to come back to the previous question, the parameters?
We are working on that in this new clinical studies, yes.